Welcome to the 10th lecture in the Robert and Russell Moody lecture series. There'll be one more on March the 6th. But tonight, we're very fortunate to have with us David Van Essen from Washington University, a neuroscientist who has been mapping the brain from many different angles. But I have to say that I'm wearing a bolo with a cardinal on it and a belt with a cardinal on it. I grew up 60 miles from St. Louis, and I'm a St. Louis Cardinal fan, not an Astros fan. And I mistakenly put on an Astros jacket the other day, and somebody said, "Are you an Astros fan?" I said, "No, I'm a Cardinal fan." And I looked at my jacket, I said, "Well, second place." So we're very fortunate to have David with us. He's going to lecture for 45 minutes or so, and then take questions as we usually do. And I've been reading some of his fascinating articles, and I'm looking forward to what he has to say tonight about mapping the brain in health and disease.
Thank you, Bill, for that introduction. Can you hear me in back?
Yeah.
Okay, I'm delighted to be here and talk to you about my scientific passions. Let's dive right in. I want to frame our conversation with some big questions to start. So how do our brains work to make us unique as a species, for better and for worse? Unique as individual with special talents, amazingly diverse amongst this group. All too many are afflicted by devastating brain disease that can occur at all ages and all stages of the lifespan. So I hope we can agree that deciphering the brain in health and disease is a grand challenge for the 21st century and beyond if our species is fortunate enough to survive. Tonight I'm going to focus mainly on healthy brains, but I'll touch upon brain disorders at the very end. So let's take a closer look at the human brain. When we see it from the side, here's the view we get from the right side. If we take a slice through the middle of the brain, we see a view you see over on the right where you can see inside the white matter and the gray matter. And if we cut right along the midline, we can see what we call a midline view. And in all three views, the dominant structure is the cerebral cortex. It contains 80% of the mass of the brain, but it only contains about 20% of all the neurons, of which there are 96 billion or 86 billion or so in the entire brain. And the reason there aren't so many neurons in the cerebral cortex is because it's absolutely loaded with trillions of synapses, the connections between nerve cells that we'll talk more about. Underneath the cerebral cortex is the white matter, the cabling that contains 100,000 miles worth of very thin fibers that convey information within one hemisphere and across between the two hemispheres by a massive structure, a cable known as the corpus callosum here in the middle. The pint-sized structure in the lower left here is the cerebellum. It's only 10% of the brain mass, but it contains about 80% of all the neurons. And it's very important for coordinating our movements and our thoughts, working along with the cerebral cortex, but I won't say much more about it tonight. Then the rest of the brain in the middle and lower regions here contains less than 10% of the brain mass and fewer than 1% of all the total neurons, yet it's incredibly important for housekeeping functions that keep us alive and well. And those rest of the brain is grouped into subcortical structures that are compact and are called nuclei as their official name. So the mystery that we want to do is to start untangling the wiring of the brain and figure out how it works. The way we're going to approach this, the roadmap for tonight, I'm going to talk about brain basics, and we'll work through a set of topics comparing brains to computers and genomes, and other topics that will work along. And then in the second half, I'll talk about the human brain more, particularly the Human Connectome project that I've been involved in for many years. And then I'll wrap it up in due course. So here's a brain on the left, and one of many diverse types of computers. Some are larger than a room, some are smaller than a fingernail. The parts list for brains that you heard a bit about already is neurons. Each neuron has a little cell body that does housekeeping functions. And then it has a receiving end that we call the dendrites that listen into communications from other neurons. It makes a decision to whether to send a signal along a single thin fiber called an axon, then branches profusely and makes contacts with other neurons locally and at long distances through these specialized communication devices that we call synapses. The signals that occur along nerve cell axons, we call them nerve impulses. We sometimes call them spikes, and they occur at a frequency of anywhere between 1 and 100 spikes per second. So if you listen in electrically to one of the signals from a single neuron and connect it to a loudspeaker, a neuron typically would go on pop, 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 pop. That's the message that the brain has to decipher when it comes to all the synaptic connections. So this is wildly different than how computers work. Computers send electrical signals, to be sure, but they send it through computer chips, central processing unit, that can do millions of calculations a second, much faster than brains. And computers, as you know, are becoming increasingly powerful year by year. We like to think that we're still smarter in some ways, though maybe we'll wake up someday and the computers will say, sorry, we're taking over because we're finding it much smarter. But for now, we say we're smarter because we're wired in a special way that we call our connectomes, the set of connections in our brain. So now I want to make another type of comparisons. How many of you have you heard of the term genome before? Okay, most of you have, at least many. The genome is the sequence of base pairs. The alphabet soup, 4 letters make up the DNA alphabet. And in the human, each cell has about 3 billion base pairs forming its genetic sequence. A mouse, amazingly, has almost as many as we do. And if you take the next major aspect of the genome, there are about 21,000 genes in the human genome. Each gene is specialized for generating a single specific type of protein. But remarkably, humans and mice have about the same number of genes. So that's the instruction kit for making a brain and a body. When a mouse uses that instruction kit, it makes a brain of less than 1 gram. When our genome makes the brain, it's about 1.5 kilograms. We have more than 1,000 times as many neurons as a mouse brain. When we talk about the anatomical subdivisions like I did before, the number of subcortical nuclei, which in a way are analogous to the genes in relation to the genome sequence, there are hundreds of subcortical nuclei in mice brains and hundreds in our brains. Not a huge difference, but some, to be sure. When we talk about the cortical areas, which I'm going to spend a lot of time on, a mouse has about 40 distinct cortical areas. The human, there's been a big question, a raging debate for more than a century. Is it close to 50 or maybe as many as 200? I'm going to propose an answer to that that comes closer to 200, but that's towards the latter part of our story. So I want to give you a sense of how I came to be here, how I became a brain scientist. And it started, my fascination with science started in high school, back 6 decades ago. I thought I wanted to be a physicist when I grew up. I knew I wanted to be a scientist. I thought it would be physics. I went to Caltech as an undergraduate, and I decided physics was too hard for my brain to become a theoretical physicist, so I shifted over to chemistry. And then one summer I read a book. By pure chance, I came across a book called The Machinery of the Brain. And I said, wow, that's what I want to do when I grow up for a scientific career. So I went on to graduate school at Harvard, and I did a PhD in neurobiology on the nervous system of the lowly blood-sucking leech. That was my starter. I proceeded to do 5 years as a postdoctoral fellow in Boston, then in Norway and England. And I studied different parts of the brain, the visual cortex that helps us see, and aspects of how the brain develops. When I moved on to a faculty position coming back to Caltech in the mid-70s, I continued working on visual cortex mainly, but other things. And I also got sucked into doing administrative tasks that I wasn't too bad at. And so a few years later, I was asked to become a department head at Washington University. That's how I came there, but I was able to continue my scientific research on what we're going to call cortical cartography, making maps, and the Human Connectome Project, and a follow-up project I'll mention at the very end. So, cartography for me begins with taking a closer look at brains of different species and different sizes. So here's the human brain again. Look how big it is relative to our nearest living relative, the chimpanzee. It's threefold larger. Our brains are 15 times larger than the intensively studied macaque monkey. That's one I'll talk about more. 200 times larger than the small monkey, the marmoset, I'll mention briefly again, and thousands of times larger than the mouse brain we've already seen an image of. So not only are there huge differences in size, but the mouse and the marmoset brains are relatively smooth, whereas monkeys and apes and our brains are highly folded or convoluted. And that requires us to make maps to understand what we're looking at and how to interpret it. The earliest maps of the human brain started a century ago when anatomists in Germany took post-mortem human brains and sliced them up, and then they stained each slice, in this case with a stain that made the nerve cell bodies purple, and so the cortical gray matter ribbon is all purple here, the white matter is still white. And if we zoom in on a small region, you can see that each one of those little purple dots is the cell body of one particular neuron, and the distribution of those neurons in layers and neurons in different sizes allow the anatomist to look closely for differences in what we call the architecture of different regions. And using this, the great German neuroanatomist Brodmann made a drawing that showed evidence for 50 or so cortical areas in the human brain. And around the same time, other anatomists took advantage of a different feature, which is that the axons I've already mentioned to you that carry nerve impulses, most of them are insulated by a wrapping that wraps around the entire length of the axon and is called myelin, and that myelin can be stained with a different chemical reagent, and it turns the white matter completely black instead of being pale white. But within the cortical gray matter ribbon, the myelin, there are many myelinated fibers extending up in there, and differences in their pattern allowed for dividing up the cortical sheet according to what was called myeloarchitecture. And another group of German anatomists, again early last century, identified an estimated 200 cortical areas by myeloarchitecture. So that set the stage for this century-long debate. How many areas are there? And we need to know, because that's the roadmap for understanding the human brain function. So we'll come back to that. But the other thing you can see here is that these maps from a century ago were incomplete. They only showed the outer or exposed surface of the cerebral cortex, and what was going on deep inside these complex folds was not shown on the maps. Well, when I started working on cerebral cortex, I was studying initially in the mid-70s macaque monkeys as an animal model, but I had the same issue of wanting to make maps that showed the whole cerebral cortex. And I used as an important analogy what cartographers of the Earth's surface do. They make flat maps. They take the spherical Earth, which has wrinkles and hills and mountains and valleys in it, and they make cuts to flatten it out, and then they make a two-dimensional map onto which can be displayed maps of geography, of political subdivisions, and tons of other information. So the same idea emerged for the macaque monkey, and this was, in some sense, naturally an idea for computers, but back in the 70s, I didn't have the computer horsepower to get it done, and so instead we literally used pencil and tracing paper to make our early maps of the monkey cerebral cortex. So this is a map of the folds, the geography, what regions are buried in sulci are shown in gray. But more excitingly, we were able to use these flat maps of the monkey cortex to paint on it a mosaic of distinct cortical areas, several dozen areas in colors that were associated with vision and lots of other areas with other functions. And each cortical area was identified based on what we knew from the scientific literature and identified according to differences in their function, their architecture like we saw in the previous slide, their connections, or their maps of other sensory surfaces. And some of these areas in this early map were very well defined. We call them poster children because V1, this primary visual area, and this little motion processing area called MT could be well defined by any or all of these different approaches. But most of the other areas on this map, we drew the borders sharply, but the boundaries were actually fuzzy, and they were debated for many years, even to this day, as to which borders are real and which ones are just artist impressions. So parcellation of the cerebral cortex into its functionally relevant subdivision, that's the key objective that we're focusing on tonight. It's a really hard problem, and we need new methods to do it. And one of the most powerful methods, particularly for humans, is magnetic resonance imaging that came around in the mid-1990s into human brain studies. So here on the left is a physical brain slice from a postmortem brain like you saw before. On the right is a structural magnetic resonance image, or MRI image, that shows white matter and gray matter in cerebral cortex, both hemispheres, and the underlying cerebellar cortical sheet. So how many of you have actually been inside an MRI scanner? So quite a few have, and I want to try to demystify it just a little bit because it's a powerful device, and I want to give you a sense of how it works, just briefly. So schematically, we have this image down in the lower right. It starts with a powerful magnet. We're going to be talking about a 3 Tesla magnet that is thousands of times stronger than the Earth's magnetic field, and that magnetic field inside the scanner is generated by a cylindrical outer region here. That's the main magnet in black in this lower drawing. And then there's inside that a set of electrical wiring that's called gradient coils. They basically cause modest changes in the strength of the main magnetic field, and that's very useful for the spatial localization that the scanner can achieve. The thing that makes all the noise, if you've been in a scanner, they're very noisy devices, and that's because there are radio frequency transmitting coils that generate what are called pulse sequences that have a very complex time course, and they cause vibrations that make the sounds. But the important thing is the electronic signals they generate, which are picked up by extremely sensitive receiver coils that are placed around the head sitting inside the scanner. So that detects the faint signals and sends the signals to computers that decode and reconstruct the images like the one you just saw and carry out many stages of data analysis that you'll be seeing as we go forward. So here's another MRI scan, a movie running from back to front. Here is cerebral cortex above, cerebellar cortex, ventricles, fluid-filled sacs inside the brain. That was a 1990s version. Nowadays, we can get much higher quality structural MRI, and we can, such as the one you see here, and we can also use fancy computer algorithms to extract the shape of the cortical sheet. So the blue and green contours here that follow the boundaries of the cerebral cortex can be used to generate a computer model, such as you're seeing over on the right here. So this computer model captures the convolutions, the shape of the original 3D brain, but we can also manipulate its shape. We can literally inflate the computer model and preserve the original shape features by showing the darker streaks here are regions that were buried in the original 3D model. So this is a powerful way for analyzing and visualizing the cerebral cortex. If you look from above at the left and right hemispheres, you can see they're similar but by no means identical in their folding patterns from just within one brain. And you can also note that we can computerized methods can allow flattening of the left and right hemisphere separately. And now I can tell you an important factoid that the size of each hemisphere is about the equivalent of one 13-inch pizza. I'm hungry actually now. I wish I could have some pepperonis, which are about the size of the individual cortical areas that we've started to talk about and we'll say more about. So that just gets you calibrated. Another hugely important and fascinating matter is that the patterns of folding from one individual to another are very different. So here's images of 4 healthy young adults shown on the left, and we can get another view, inflate them, and show these maps of the folding pattern, and now we can see that in some regions, like right here on the top, there's a fold that's called the central sulcus. That's important because it separates the regions involved in touch behind and regions involved in controlling movements right in front. The central sulcus is always there consistently, but in other regions like this region farther back into the down a bit, that's a higher visual area involved in processing motion and other features. And it's folded very differently from one person to another. And amazingly, that's true even in identical twins. So lo and behold, we chose these as one pair of identical twins below, another pair above. In both cases, the identical twins, they had the same genetic instructions during development, but the folding pattern came out wildly different. There is some heritability, but what is most striking is the differences. Another important characteristic is that the cortical areas that we're continuing to talk about vary in size across individuals. So the red outlines in the lower right here are 3 different brains showing in red the primary visual cortex, like we saw in the monkey, now we're seeing it in the human, at the back end of the brain. And the point is that if in some individuals, the area V1 is much smaller, in other individuals it's larger by a 2-fold range. And also the boundaries of this area are not consistently related to the cortical folds that we've been talking about. So that poses some interesting challenges, but it also poses a fascinating question. What is it that makes the cortex fold? Well, we know when cortical folding occurs. In this schematic, you can see between the beginning of the 3rd trimester and the end, a lot of folding takes place. And if we look at actual data, here are MRI scans at different ages from preterm infants, and you can see at the beginning of the 3rd trimester, there's hardly any folding, and the surface model confirms that. By the end of the gestation, the cortex is highly folded, almost as much as it is in the adult. And that's repeated in these figures. So at the term equivalent, when normal birth occurs, the brain is almost as convoluted as in the adult, but it's only 1/3 the total size. But when that action occurs, there's a lot of growth of the white matter underneath. And I started thinking a bit more about that. Literally, one night in my living room, I was pondering some observations that we've made in my lab, that the connections in monkey brains between the primary visual area you heard about, V1, and a neighboring area, V2, those connections between those 2 areas are established by axons growing out and connecting the 2 sides right around the time that the cortical folding occurs and a gyrus forms in between. So the light bulb moment for me was maybe that wasn't a coincidence. Maybe there's actually a cause and effect here. And more specifically, I hypothesized that tension, mechanical tension along the axons connecting 2 regions could pull strongly connected regions closer together, forming a gyrus in between. And the opposite kind of fold that we call a sulcus, by this hypothesis, was the outcome of a losing battle where there wasn't enough connection strength to prevent them getting pulled farther apart. So this one hypothesis can account for the consistency of folding like you see in the central sulcus, and for the variability in other regions that are balkanized little patches of areas of different size and connection patterns. It remains an attractive hypothesis 2 decades after I proposed it. It's still unproven. It's actually controversial. And it's probably only one of several mechanisms that I won't try to elaborate the others, but it's common in development mechanisms that more than one factor contributes to something as important as things like cortical folding. Okay, one more important set of points about that we learned from studying animal models is cortical wiring or connectivity. And if I can get the slide to advance, that would help. So this brings us into the realm of what we call connectomes. A connectome is a comprehensive map of the wiring of all the neuronal connections. However, I have to put it into quotes because we can't actually get a true start to finish map of all the wiring. There is a world that we call the microconnectome world that uses electron microscopes and other fancy methods at very high resolution to reconstruct every neuron, axon, dendrite, and synapse in exquisite detail, but only for tiny amounts, far less than a cubic millimeter. Other methods allow us to probe what we call the mesoconnectome, the middle range where we can use, in animal models, pathway tracing techniques to identify local and long-distance connections. We're going to come back to that in a moment, but I want to introduce the third player in the connectome realm. That's what we call the macroconnectome, that is particularly relevant for the human brain. It lets us, with the Human Connectome Project, analyze the wiring, but only down to the level of a cubic millimeter or two, so much coarser than these other methods. So we're going to come back to that, but I want to tell you a little bit more about basic principles of wiring in animal models. So here, let's take a mouse brain. You've seen a view like this before. Here's our schematic model of a neuron and an axon and a synapse. Let's take a look at a real-world one. There are now powerful methods that allow us to view in their entirety all the dendrites and axons of a single neuron. So this is just one neuron in the region of a mouse brain that controls movement. Here's another neuron in the mouse brain that is involved in vision, area V1. And this one is cool because we can take a movie and take a cruise to see close up the dendrites and axons right around where the cell body is, then long axons going major distances and branching and hitting targets and making lots of synapses. So this is the anatomical reality for every one of the 90 million or so neurons in a mouse. And that's how the wiring is used to connect different cortical and subcortical areas with one another. So we now have much better maps of the arrangement of cortical areas in the mouse. So on the lower left here, you can see a mosaic and faint white outlines that show the map of 41 cortical areas that have been identified in recent studies. In the middle panel here is a pathway tracing experiment that can be done in the mouse. And the idea is to put a tiny injection of a fluorescent molecule into one area, in this case area V1, and that fluorescent molecule is picked up at the synapses and it is transported, it is carried back in the backwards or retrograde direction back to the cell body. And every little yellow dot you can barely see here is one cell body that has an axon projecting all the way to V1. So there's thousands of labeled neurons that are projecting in a connection with this one injection. And if we systematically analyze all of these, we can get what is called the connection matrix. Each little box here represents the connection strength between one cortical area and another in the way that's been analyzed here. It's not a complete map of all the wires, but it shows that there's extremely rich, what we call dense connectivity throughout the mouse cerebral cortex, much more than people would have guessed some years ago. This repeats what you saw for the mouse. Now if we take a look at non-human primates, the marmoset has been intensively studied. It has a connectivity matrix that you see in this middle section. The macaque monkey that we've talked about before has a connectivity matrix that's shown here. We're not going to get bogged down in the details. The basic point is that there are thousands and thousands of pathways connecting different cortical areas. They range dramatically in their connection strength, and they reveal that the wiring of the animal model brains are far more complex than people had realized. Why are we talking about this? Because we want to know if it generalizes to human brains and cerebral cortex. If we want to explore that, we can't use these invasive tracer methods that work in animal models. So instead we turn to in vivo imaging using MRI. And we're going to talk a little bit about what has emerged with what's called the Human Connectome Project. So first, some general words about what this project was. It was a 6-year project to acquire data on brain structure, function, and connectivity in healthy adults, in fact, twins and their siblings. We used powerful new scanners, powerful new pulse sequences developed to help improve our data quality. We got 4 hours of imaging data, 4 one-hour sessions from each of our volunteers, and we were able to get 1,100 subjects to be successfully scanned at this scanner. And we also got behavioral data and other information about each of the participants, the healthy volunteers. Then we analyzed the data intensively using the processing that computers need to do to clean up the noisy data and align the data across individuals and visualize the data in fancy ways and carry out advanced analyses, one of them involving parcellating the cerebral cortex that we'll come to in a short while. Then we followed the advice of our funding agency, the National Institutes of Health, and we shared the data widely. So literally more than 10,000 investigators have already tapped into this rich public data source, and over 600 publications and growing week by week have been citing this data as part of their scientific discoveries. So what have we learned with this? Well, let me set the stage by reminding you that the classical postmortem anatomy allows us to look at cytoarchitecture, like you see on the left, and myeloarchitecture, the differences in myelin staining on the right. The myeloarchitecture turns out to be something we can actually probe in living humans, not waiting for postmortem analysis. And this was inspired by my very talented graduate student a few years ago, Matt Glasser, who when he came to my lab had noticed that in MRI scans, a typical scan was like you see here, where the white matter is white, the gray matter is gray, and but in some regions, the gray matter is a little bit brighter than average. In other regions, it's a little darker than average. This is what is called the T1 weighted image, setting the pulse sequences of one particular way. A short while later, in the same subject, in the same scan session, was given a T2 weighted image scan, and the region of gray matter that had been brighter is now darker. The one that had been darker is now brighter. So they're complementary, and one can gain sensitivity by taking the mathematical ratio, the ratio between the value at each voxel for what the T1 value was and the T2 weighted value, and then colorizing it and mapping this from the slices onto the cortical surface that we've seen models of before. So this is what we call a myelin map of this individual subject, and we call it that because there's a striking correlation between the red regions and yellow regions being heavily myelinated when compared to the postmortem data that I've shown you in a previous slide. When we do this for not just one subject, but many hundreds of subjects, and align the data very carefully across individuals, we get what we call a group average myelin map that is much cleaner and crisper, and we can look for subtle changes in the intensity that are consistent to suggest areal boundaries between different cortical areas. We can take from the same group of subjects the maps of the thickness of the cortex and see that some regions in dark are very thin. That's the touch-related regions of the cortex. The regions controlling movement are much thicker, but they're both heavily myelinated. So we get complementary features informative about the subdivisions we want to analyze just from looking very carefully at high-quality structural data. That's good, but it's not enough. It doesn't give us a complete map, particularly in these lightly myelinated regions here that are involved in higher cognitive function. And those we really want to dive further into. And so we turn to functional MRI to help with that. And Deanna Barch at WashU spearheaded this project. I'm just going to whiz through quickly to give you the flavor of what is functional MRI about and what does it tell us. In each of our subjects, they spent an hour doing 7 different tasks. One of them was listening to stories in the scanner compared to alternating with doing arithmetic in the scanner. Another cool one was to just look at images and look at the image on the left and say, are those triangles hugging, kissing, and dancing? Or are they just randomly moving past one another as happened in other episodes? And that was a judgment of what we call social cognition. The third task I'll illustrate was to look at a sequence of images flashed up one after the other and say, is the image you're just looking at the same or different than the one you saw 2 images previously? It tests what we call your working memory. And we can display the activation patterns from the MRI scanner on brain models of the type that you see here. This is just the underlying structure. And now when we say what parts of the brain are turned on in red and yellow during listening to stories, and you can see that there are regions of the temporal lobe and the frontal lobe that are turned on. Other regions are turned off. You can see the left hemisphere is turned on more, but the right hemisphere is also pretty active. If you take the next task, remember that social cognition task, that turns on a different set of regions, nearby in some cases, but distinct and complementary. If you take the third task that I showed, the working memory, what was that image 2 previously? That taxes your frontal lobe cortex, what we call prefrontal cortex, and a region back here in the back that we call the parietal lobe. And so, in general, each task engages a complex over network that overlaps partially with those involved in other tasks. This doesn't give all the answers we want, but it gives you a hint of how your brain actually works when it's doing a task it's instructed to do. What if you're told to do nothing in the scanner except just look at a blank screen or that has a small crosshair in front of it? Well, this is the brain at rest. This is one of our participants who is not doing anything instructed, but may have been thinking about what's for dinner, what should I wear tomorrow, what about that snoozer of a Super Bowl, and so on down the line. So this is what we call resting state fMRI, functional MRI, and we can interrogate that by looking at the time course, the fluctuations in this signal at one region and comparing it to the time course in another region and asking, are they strongly correlated going up and down together or going in opposite polarities or just uncorrelated? We call that functional correlations, and we translate it as functional connectivity because we think it's an indirect measure of the actual wiring. Strongly correlated regions are likely to be strongly connected. And we can do this for all 90,000 by 90,000 possible patches of gray matter, and then we can interrogate these maps and say, for any one region, like region number 1 here, who is it correlated with strongly as in yellow or not at all in blue or green? And we can do that for other regions, and we can use this information of what we call functional connectivity as an important, actually the most valuable of our different types of information for divvying up the cortical sheet into what we call parcellation. And so that's what we'll touch upon in this next little subsection. I remind you of where we left off a century ago. Brodmann said there's 50 areas by cytoarchitecture. The other German anatomist said it's closer to 200. In the intervening years, particularly in recent years, people using just functional, resting state functional connectivity came up with maps that are better than what we had before, and there have been several of those. But it turns out that just using one type of information, what we call a unimodal parcellation, doesn't get us as rich and complete a picture as we want. So Matt Glasser and I decided to focus intensely on the Human Connectome Project data and use as many modalities as we could bring to bear on it, and to generate what you see here. This is an parcellation generated from an average of 210 subjects. We call it the HCP multimodal parcellation. And you can see it's a mosaic of 180 areas that are strikingly similar between the left and right hemispheres. And we get a functional signature by showing in green the regions that are involved in touch and movement, in blue the regions involved in vision and sight, in red the regions involved in hearing. In bright white and light colors are regions that are more active in general cognitive regions involved in activations when you're doing a task. The dark regions are interesting because they're actually turned off when you're doing a task. They're more active when you're just introspecting and freewheeling. We're going to come back to that because it's important clinically as well as fascinating. We gave each of these cortical areas a label, a name tag. 83 of them were drawn from the previous literature. 97 were what we call newly identified areas. Matt was able to train a fancy algorithm to take the group average data and map it back to individual subjects so we can identify these same areas in nearly all subjects for nearly all areas and show that they vary in size and shape and location in ways that I touched upon previously. We can show that these areas are grouped into what we call functional networks based on the resting state data. There are, just in brief, there are 12 major areas or networks, each shown in a different color on the map, and the connectivity patterns that derive from that in this complex mosaic, this matrix here, are shown. There's one I want to draw your attention in particular to, this reddish region, this set of parcels at different locations in different regions, is what we call this default mode network or task negative network. Hang on to that, we'll come back to it towards the end. So one of the broader lessons that emerged from all this work on the Human Connectome Project is we had a new way, a new game plan for recommending how major studies go going forward. And there are 7 core tenets to what we call the HCP style neuroimaging paradigm. And just in very briefly, get as much high quality data as you can at high resolution in space and time. Avoid distortions and get rid of noise as best as you can, because that's a major confounding factor. Respect the geometry of the cortical sheet and other brain structures. Align data across individuals as accurately as possible, even in the face of the diversity of folding patterns. Analyze the results using an accurate parcellation of the type that I just showed you for the cerebral cortex. And then freely share the data, not just the data that came into the analysis, but the advanced data analyses. So this is now happening not only in what we now call the young adult HCP, the original one that studied people from 22 to 35, but we've now bookended that. We have studies going on at WashU and elsewhere, what we call the lifespan projects in aging and development, to cover up to beyond the century's worth of age, and from 5 to 21 in the development project. There's another exciting project that's even vastly larger in scale, the so-called ABCD, Adolescent Brain and Cognitive Development Study, that's taken 12,000 kids, starting at age 9 and 10. They scanned them once already, and they're going to follow them for a decade and get a really deep dive into what's going on in the teenage brain. What a mystery it is to many of us. There's a baby connectome project and a developing human connectome project that are going from prenatal up to age 5. So we're going to cover the brain development across the entire lifespan in healthy individuals. What about brain disorders? Well, the good news is that there are 14 major projects, again funded by the National Institutes of Health, looking at brain disorders using this HCP-style paradigm to study neurodegeneration, dementia, depression, psychosis, epilepsy, and blindness. That's just a starter kit of all too many disorders, but it will give information of incredible value. So in my remaining moments, I want to talk a little bit, just briefly, to give you a context of thinking about brain disorders. Most everyone in the room either has had or will have in your lifetime some brain disorder. And everyone probably knows of somebody, a friend or a relative, who's facing that kind of severe challenge. It's incredibly devastating in social and economic impact. And it's challenging because there's so many ways the brain can malfunction and misfire. And at present, we have few brain disorders that can be cured, and the treatments in general are far from satisfactory. Why? And the basic reason is we still don't understand what's gone wrong or how to fix it. What do we need? We need more basic neuroscience in animal models as well as in humans. We need to improve the technology even further. We need translational, bench-to-bedside translation of discovery into improved treatment, and we need to implement improved diagnosis and treatment. So how are we going to do that? I'm going to touch very fleetingly on 3 examples relating to development, dementia, and depression. So for development, if we look at preterm birth, this is two healthy infants at the time of scanned at birth. And here are 4 brains from babies who were born very prematurely, 26 weeks or so, and then scanned at term equivalent. And you can see that there are regions, particularly in the lateral temporal cortex, this lobe down here, particularly this individual, that are far from developmentally appropriate. So there's a selective vulnerability in the intensive care unit to proper maturation of the brain and cerebral cortex. We hope that improved methods will let us dissect that further and give better methods of diagnosis and treatment. Aging is something that, of course, occurs in this room as we speak. As brains get older, even healthy brains change. There's more atrophy, larger ventricles, and more space between the folds. But more serious and severe is the dementias, particularly Alzheimer's disease. So remember that I told you to try to remember the default mode network in red in this network I showed before. Here's another demonstration of it from a different study. The default mode network turns out to correlate with regions of high energy metabolism, what's called aerobic glycolysis, using oxygen to burn up sugar. But what's important in the right side here is that in Alzheimer's disease, there's abnormal metabolism concentrated in the default mode network regions that we've just introduced. And even worse, there's something called amyloid that is a hallmark of pathology in the demented brain, and that amyloid is concentrated early on in this default mode network region. But it can be seen, the amyloid and other markers soon, we hope, many years before there's clinical symptoms. So it gives us hope of having biomarkers that can be used to test are drugs actually helping to slow or halt the onset or progression of Alzheimer's. That's the hope. And the last quick example is depression. It's very common, clinical depression. Drugs often don't work. In extreme cases, putting electrodes into the brain, into this little patch near the front of the brain, and electrically stimulating, deep brain stimulation can help. But it's an invasive procedure and not frequently used. There's hope for the future that something called transcranial magnetic stimulation, putting a magnetic field pulses just right outside the brain, can help, at least in some cases, with improving depression. But neuroimaging is coming into play because a recent study has shown 4 different types of individuals according to their functional MRI networks, and those individuals differ in their responsiveness to the transcranial magnetic stimulation. So it's hopeful that this will come into play as a better way of analyzing and diagnosing and providing more sensitive, personalized treatment. So, understanding the human brain is a grand challenge. The Human Connectome Project has given us a lot of progress already, and it's not over yet in terms of what will be discovered. We're getting more insights and discoveries. We're not going to get a human connectome all the way down to the level of individual synapses for the whole brain. That's just not in the cards. But we can hope that the HCP-style approach that I've articulated is going to accelerate progress in understanding, diagnosing, and treating a variety of brain disorders. So one last analogy, we talked about maps. Recall that Earth maps started in quaint forms, as did brain maps a century ago. Book atlases proved very useful for a while. Brain atlases also had their heyday, but now we've moved into things like satellite imaging for the Earth and MRI, functional and structural, for the brain. Google Earth has absolutely transformed our understanding, our way of navigating the Earth, and we hope that the world of connectomics, as it continues to progress, will accelerate our understanding of the human brain in health and disease. So everything I've talked about was a team effort, an incredibly talented group of individuals. Matt Glasser, I've mentioned, Deanna, Camille, Dan, Steve, and people in my lab, and the funding agencies. Your taxpayer dollars, we hope you think have gone to good use in this effort. So I'd be happy to answer questions. Yes. So I'm not an expert in this, but I'll do my best. Executive function is that set of processes that make you make, hopefully, good decisions. What are you going to do? What are you going to execute? What are you going to have for breakfast? What are you going to decide to go to college? All those things require planning, forethought, integration of lots of information, and to execute good decision-making. It used to be thought that it was more lateralized, but the more, at least my bias from our emerging understanding, is that the left and right hemispheres are actually much more symmetric than was thought back in the days when left brain and right brain were kind of the mantra. And there are still asymmetries, and there are still striking effects of lesions in one side versus the other. But I think by and large that the two sides work in a very coordinated way, but in ways that are honestly still mysterious, especially for these exotic things like, how are decisions made? There's a lot of intense work being done, but we can't say that we've figured it all out yet.
Question. You had several slides that showed the aging of the brain and atrophy as the brain typically gets older. Are there things that can be done to slow that process?
Exercise. Exercise your body, exercise your brain. I think as a non-clinician, but one who's trying to learn about it for my own health as well as others, using it can help you keep from losing it. But you're not going to stop it. I think some things are inexorable, but you can keep your brain healthier by keeping mentally vigorous and active, and physically so as well. I think so far that's better than any pill that's on the market. Any questions from the kids in back? We're going to have a pop quiz. How many of the 180 cortical areas can you remember? Questions?
I have a question for you.
Oh, here's one.
Where are you headed next with the brain research?
I'm really keen to see these lifespan projects and these other projects that are currently underway actually yield some pay dirt. We're trying right now to transition from collecting the data to making the data freely available to the community of investigators, but that includes our own team. And so we're going to be diving in and trying to answer some of those questions, but most of the discoveries will come from the community latching on to these enormous resources. But it's going to take a few years to go from acquiring the data, releasing it, and people diving in and doing the sophisticated analyses that take a lot of hard work as well as hard thought about how best to characterize. And I'm really hopeful that these, what we call the disease connectome projects, will yield deeper insights by virtue of the higher quality data and more careful approaches to analyzing so we can get better understanding, for example, in dementia or schizophrenia, what is actually going wrong in what is another thing just to mention. I'll get back to you guys, but one thing I want to mention, just many of these brain disorders, schizophrenia, autism, depression, they are not a single disorder. They are a spectrum. And that makes it all the more challenging because there's not going to be just one thing that's wrong in an autism spectrum disorder. It's going to be a portfolio that makes it much harder to decipher, but also extremely important to decipher at a level that can allow for essentially personalized treatment. That's why the one example I whizzed by on depression, 4 identified subtypes of depression based on brain imaging are clinically relevant to recommending which treatment is more appropriate to try. That's just the starter of what I hope will transform personalized medicine. To what extent does genetics play a role in brain deterioration? A large extent, at least in many cases. So, for example, there is a particular distinctive type of what's called ApoE4 is a particular variant of a genetics, a gene that everyone has. But if you have that variant, you're at much greater risk of having early onset Alzheimer's. That's one example. Unfortunately, most examples are turning out to be much more diverse and complex. For example, autism is unquestionably has a very large genetic contribution, but intense efforts to find the autism gene have made it clear there's not going to be any the autism gene. There's, I think, 100 or more strong candidates that are implicated, and having the wrong collection of genes may increase your susceptibility, but it's still early days, in part because it's just so incredibly complex, the way the genotype interacts with brain development and maturation. But another thing to make in a general way is that genes, by and large, they don't drive the outcome. It's genes interacting with brain development and the environment. So even if you have what you might think are bad genes or risky genes, having the right environment or right environment for your children or at various stages may make a huge difference in the outcome. So it's, in a sense, gives reason for hope and not feeling we are slaves to our genetic instructions yet. Did you have a question?
I had one about gender differences. We see women lie differently. Is that still something people believe at birth?
So that's a wonderful example of a question that is already amenable to intense analysis with the data sets, the Human Connectome Project data sets that we've delivered and released. I'm aware of a few studies that have started to probe that, and there are certainly gender or sex differences in brains. The most notable is that female brains are smaller. One can say they're leaner and meaner, because in that case, smaller doesn't mean worse or better, it just means different. I think the short answer is really wait and see. The 600 studies, I think it'll be studies that come out in the next few years that will actually dig into that more closely. And it will also, with these lifespan projects, we'll be able to look at when do these differences appear and how do they, they're not likely to be just a single trajectory. In fact, things like puberty have profound effects on brain maturation. Menopause has major effects on brain function. And so all these things are going to be much better understood in the next 5 to 10 years through the progress coming from these large-scale projects. Yes?
Is patient MRI anywhere useful for these studies, or is it also part of that database?
Sorry, is what MRI?
Patient, so, I mean, when somebody goes in for a brain scan, does that data go into the database as well?
No, it does not. There's several things to add. One is standard clinical information is protected health information, and so unless a consent form has been signed in advance that authorizes sharing of data, it's restricted. There is intense interest in making better use of what comes in through the clinic to inform the community about clinical issues and problems. And there's a lot more going on than I know any details about, but I think the field is changing to a desire to find the sweet spot that protects privacy when privacy needs to be protected, but to allow appropriate sharing of clinically relevant data for research purposes. But it's a minefield basically to navigate, and I'm certainly not expert in it. But the other thing I would mention is that the methods improvements that we've been talking about here are just starting to feed into improved clinical scanning. So when a patient goes in and gets a routine scan on a weaker 1.5 Tesla MRI machine using conventional methods, it may help the initial clinical diagnosis, but it may not be powered to capitalize on the insights that we're gaining from these basic research studies. So it's a moving target, and I think clinical medicine is one of the many ways it's going to transform in the years and decades to come.
I have another question. You talk about the lifespan. Have you done anything with the developing fetus and the brain development prior to birth?
Great question. There are two parts to it. One is I was involved in a collaboration on these preterm infants where when an infant is born, say at 26 weeks and is kept alive, they're also kept in the intensive care unit, and they can be scanned either for clinical diagnosis or just for research purposes. And so that's a way of probing those third trimester periods of maturation while the infant is out of the scanner. But an even more exciting advance, I think, is in this British Developing Human Connectome project. They have figured out ways to scan babies in utero by putting the mom in the scanner and then tolerating the fact that the kid doesn't sit still. It's wiggling around, kicking and wobbling. And so the challenge is a technological one, but they've figured out how to compensate for the rapid movements and align from one movement period to another. So that's starting to yield some fascinating information about healthy maturation in utero. Everybody ready for some pizza?
I have one question. This is unfair to ask you this, but with all of the research that you've done, what is your personal view of the production of consciousness? I mean, you must have an opinion about it. I'm not saying that.
I've talked to Christoph Koch, who was one of your first speakers here, I think, who is not only fascinated by consciousness, but thinks hard about it and has ideas about it. For example, that one structure that I didn't mention called the claustrum is a particular anatomical intriguing structure that because its neurons branch especially profusely, Christoph has proposed that it is centrally involved in consciousness. I guess my short answer is that's beyond my pay grade. I don't know how to think about it in a scientifically intelligent and productive way. So while I'm fascinated about it, I'm happy to muse about it. If you give me a beer, I might muse more. but I don't know how to, I can't get my head around it in a scientifically deep way. So it's a fun topic. Is it actually accessible? Sometimes I respond to questions of that type by saying, can the human brain actually understand the human brain? Are we that smart, even with the help of the supercomputers that are, doing a large part of the grunt work, in a sense, for the analyses the pretty pictures that I showed you? And I don't know. Response number one is, well, what about chimpanzees? Can a chimpanzee understand a chimpanzee's brain? And I think everyone would say, well, obviously not. So our brains are 3 times bigger. We're much more clever, but are we so much more clever that we can actually go all the way to a deep, satisfying understanding of something as esoteric as consciousness? And my answer is a strong maybe. I wouldn't bet the farm on it, but I sure hope, I know we have far from exhausted what our cleverness can yield, but whether we'll max out at some level where we just say we're too befuddled to get our hands around that, I don't know. Yes?
The electrical activity of the brain is almost more like software, while the brain is almost like hardware.
You're saying the electrical activity is more like software and the brain is like hardware. In a real sense, that I think is a good common an analogy. So the software is the manipulation of how information flows constrained by the physical hardware. In the brain, the signals and their very dynamic fluctuations, much more rapid than the fMRI signals that I showed you, which are slowed down by what's called the coupling between real activity and changes in blood flow. For the electrical activity, it's extraordinarily dynamic, and it's intensely studied in ways that I had no time to capture. But it is, in a sense, like the software. The big difference, if you want to say, what is the biggest difference that I didn't mention about brains versus computers? One is that computers have CPU specialized hardware that does the computation. It does the arithmetic, it sent the answer and puts the answer in memory. And so you can have gigabits or terabytes worth of memory, but the computation is done in a customized computing device. In our brains, the computations are done at synapses, and the memories of what was just computed are essentially stored at synapses as well. And so that's yet another of the profound differences in the way it works. So to say the human brain is a computer, yeah, I think of it that way. I just think of it as wild, even more so than I would guess 10 or 20 years ago.
Have you read Norman Deutsch's work on neuroplasticity on that brain?
I have not read Norman Deutsch's specific work on neuroplasticity. I know bits and pieces of plasticity from other studies. What is it you're thinking?
I mean, you're mapping the brain to function, and he talks about how people have had certain parts of the brain destroyed that can regain function somehow for rehabilitating their brain somehow. And I didn't know if you had an opinion.
I have a profound respect and sense of awe at how much plasticity there can occur in the brain, even most by far at early ages and progressively less. So my brain is not nearly as plastic as yours, but I hope there's still a little bit left. But certainly one of the lessons is that the brain can recover from damage or insult by far the best at young ages. And there are cases, for example, of kids with epilepsy, where the treatment is to remove half the brain. Scary thought, but when they grow up and are far better functioning than they would have been with their chronic seizures, it's a good solution, but it requires tremendous plasticity. And so there are many examples of that, and there are obvious examples of where even in adults after traumatic brain injury, recovery can occur and rehabilitation is gaining increasing respect because it's hugely important in shaping brain circuits to adapt as efficiently and effectively as possible in response to whatever insult or injury has occurred. So that's another exciting area, as well as, if we can better understand what causes plasticity or what causes it to turn off, there might be magic drug treatments or pills that would make you more plastic in a good way. But there are some tantalizing basic science discoveries, but it's still a long ways off, I think.
Last call for questions. Okay.
Do you have an opinion about, like, reading Roger on story about L theory and amyloid and research about
L-serine is an amino acid. Is it meant as a
correlated with not forming amyloid in genetically defined individuals? That's not
So although I did talk a bit about it, I'm far from expert. I know that there's intense efforts to find drugs, treatments, medications that slow or halt the onset of amyloid and the tau protein, the two killer proteins that used to be, it was one or the other. I think now there's growing recognition they're both bad guys in this mix. And which one is going to be clinically more responsive, I don't know. I simply don't know whether this particular one you mentioned, L-serine, could be helpful, could be false positive. So. Thank you for your patience and your curiosity.